Hello, good morning. So my name is Aravi Samuel, one of the organizers of this meeting, and it's my pleasure to introduce Professor Huda Zoghbi to give the first scientific talk of this workshop. So as you know, one of the key questions about the Brain Activity Map project, the Brain Initiative, and so on, is the interplay between basic science and translation, perhaps the interaction between different agencies like the NSF and the NIH. And so the organizers, the sponsors, and I quickly agreed that the thoughts and perspective of Professor Zoghbi would be invaluable in grounding this conversation, and we're delighted that she was able to be here today. So Dr. Zoghbi actually began her career as an MD, as a pediatric neurologist at Baylor University, where she first got interested in basic science and laboratory research. And really through the course of a storied career has made numerous discoveries that led us to key mechanistic insight into neurodegenerative phenomena that really illuminated clinical pathologies in a beautiful way. Her group identified the genes that give rise to Rett syndrome. In the study of ataxias, I think in children, she elucidated CAG repeat disorders, which are now recognized to underlie a number of pathologies. Lastly, Professor Zoghbi, these achievements have led to her receiving numerous accolades. She's an investigator of the Howard Hughes Medical Institute, a member of the National Academy of Sciences, and the feathers in her cap are perhaps too numerous to count. So it's really our privilege to have her here, and please help me in welcoming her. Thank you.
Thank you so much for the kind introduction. I should say I'm at Baylor College of Medicine, not Baylor University, just for the record, they're different places. I'm truly honored to be able to participate in this meeting. In fact, all of us here in this room are enjoying an amazing privilege, and we few get to shape a momentous initiative, and I, for one, feel the weight of this responsibility. We must remember the interest of young scientists and junior investigators who really don't have a voice today in shaping this initiative. Our trainees and these young investigators' careers and their research paths will be disproportionately affected by this project and for the decades to come. So I didn't really have very clear instructions what to do with this meeting. I could have stood here and talked about my science. I could have done anything possible. So I tried to glean what people want me to hear, to say, and I really couldn't get a very clear instruction. So I tried, and I hope you will bear with me. Here's my goal this morning. My goal is to warm us up for a crucial conversation that will take place over the next day and a half. I want to encourage us to ask hard questions and to think carefully about the consequences of projects of this magnitude. What are the consequences on discoveries and knowledge? And also, what are the effects of such projects on the practice of science? The Brain Initiative has often been compared to the Human Genome Projects. Now, we all know that the brain is orders of magnitude more complicated than the genome. But before moving into some of the concrete discussions and ideas about the Brain Mapping Initiative from the perspective of neuroscientists, I thought it would be really good to examine 3 points of comparison between this initiative and the Human Genome Project. So, first, the Genome Project had a concrete goal. I was a first-year postdoc attending a Cold Spring Harbor meeting on molecular biology of Homo sapiens in June of 1986, when Jim Watson expressed strong support for sequencing the human genome. He unwittingly launched what turned into a heated debate about the merits of the project. I imagine we will have some debates during the next day and a half. On the one hand, the project had a very clear finite objective: to read out a few billion base pairs. We knew what to do with the results, and there was a clear endpoint. On the other hand, the DNA code was just the beginning of understanding genes. Many people wondered, what would sequencing the genome deliver? Who would benefit? Is it worth spending all this money on sequencing junk DNA? Some overly optimistic types predicted an immediate revolution in clinical medicine. Clearly, that was before we realized that figuring out the DNA is just the very first step toward understanding gene function. That was before we really appreciated the complexities of the genomes, epigenetics, small and non-coding RNA, and so on and so forth. But at the same time, there have been remarkable advances. You heard that we found the Rett syndrome gene. I will tell you, this is a sporadic disorder, one in a family. Cloning that gene took my lab 16 years. Today, you can do it in a few days. That's the difference. As a child neurologist, whenever I was faced with a complicated case, a challenging case, we have to find the cause of the disorder in this child, and that's also sometimes recurring in families. We had to order tens of tests, many invasives, thousands and thousands of dollars. I can tell you today, any mystery case we've encountered in the last few months, we order exome sequencing and we get the result, an answer in 30 to 40% of the cases. So it's been really revolutionary in the realm of making diagnoses. Moreover, sequencing the genome from other species was transformative, as it uncovered the cross-species conservation of so many genes and pathways. This has been crucial to really understanding not only pathways, but for some cases, developing interventions. Then came the variances in the genome. We discovered all the differences in our genomes, and that led to the sequencing of the 1,000 genomes, which in turn really revealed tens of thousands of single nucleotide polymorphisms, whose effect remains to be determined. And that's where you will hear me later arguing we really need to understand function in addition to sequencing. Not to mention the epigenome, whose metastability serves at the interface between the genome and the environment, and which we're only beginning to understand. So, why do I tell you all this? I say all this because I'm really not troubled if we cannot predict the outcomes of the BRAIN initiative. However, we do need clear, concrete goals and milestones. So that's really the point of this first comparison. The second point of comparison has to do with technology. Technology was a big part of sequencing the genome. In fact, some scientists who were in favor of the project argued nevertheless that we should not jump into massive sequencing using the technology that was then available. They thought that the effort should be devoted to developing better technology before launching the project in earnest. Fluorescence labeling and the capillary sequencers were in fact huge technological advance. Yes, it was still Sanger sequencing, so the basic method didn't change until more recently, but it's hard to argue that technology development was not a major part of the genome project. In addition, Venter's use of genome-wide shotgun assembly was a revelation. It came mostly from improvements in algorithms, longer read lengths, and computational power. The benefit of the influx of bioinformatics due to the genome project has spread well beyond genome science. I propose we also need to think about what technology needs to be developed and what proportion of the funds need to be set aside for technology development. But of course, we cannot decide that. We cannot decide what to fund and how much to spend until we know what we want to measure. The third point of comparison has to do with the effects on the sociology of science as a practice. With all the benefits of the genome project that I just talked about, it did have one major side effect. It ushered in a period when biomedical research became mostly about sequencing, and more sequencing. Functional studies, sophisticated biochemistry and physiology, and the creativity focused on developing new technological advances in these disciplines faded for a while. Thankfully, functional studies are starting to have a comeback. I would argue that the impact of the genome project might have been far greater if the planning of the project dedicated separate funding for high throughput functional studies. maybe we would have had gains beyond molecular diagnoses. And this is something we need to think about. So before we start to discuss the goals of the Brain Mapping Initiative, I want to stress that we should guard against the tendency to let one paradigm dominate biomedical research. We cannot have one technology taking over neuroscience. We need to be sure that the project leaves room for new and creative ideas that none of us in this room can think of today. We also have a responsibility to make certain that we do not take away from investigator-initiated research. We're meeting today under the shadow of the sequester. And at a time when science as a whole is under attack, we need to protect the ability of scientists to pursue the questions they deem most interesting and important. So with these comparisons, I'd like now to move on and switch gears to share my thoughts about the BRAIN initiative. I'm not here to suggest one goal or another. This is much better done collectively. But I thought I would share some thoughts for us to consider over the next day and a half. There has been a lot of talk about recording from every neuron in a circuit so that we can understand how the circuit functions. As we consider such an endeavor, however, we need to think about what it will take to decipher an emergent circuit state. To understand how the brain works, I believe we need to know the following. We need to know all of the components of the brain, not just neurons, and we need a much better idea than we currently have about what defines them at the molecular level. We need to know how these components connect and communicate. We need to figure out how to measure such communication. And most importantly, we need to know how brain activity varies with experiences and external stimuli. Let's start with the components of the brain. What molecules are present, at what levels, and in what spatial distribution? This will let us figure out how the cells got the way they are and maybe how we can reprogram them. We're not even close to knowing all the different cell types and what defines them. We all know glia outnumber neurons, yet we have barely a concept about their diversity. While we need better technology to define the molecular anatomy of the various brain cells, we have a framework to permit rapid progress in this area. The Allen Brain Institute has laid a really nice foundation for such efforts, so did GenSat and other projects. This is something we can start doing right away, and the data can be disseminated so that the greater community can begin to put it to use. This is one example of a concrete goal that would require some investment in refining technology. So clearly we're not there yet to really figure out every brain cell and its molecular anatomy. But we have a framework and we can improve and refine the technology so that this would benefit the entire field and be widely used. I'll share an example with you from my own work. As you heard, we found that mutations in a gene that encode a protein called MACP2 cause Rett syndrome. This is a terrible disease. This is childhood disorder where children begin as healthy, normal kids, and somewhere around 2 years of age, they will lose social skills, they will lose all language, and gradually lose all motor function, have autonomic abnormality, and pretty much have every dysfunction of the nervous system you can imagine. So with the protein is expressed all over the brain, but with such wide-ranging symptoms, we thought, well, maybe to dissect this disease, we can start taking this gene from few cells at a time. When we took out this gene from inhibitory neurons, we were quite surprised to find that just taking it from less than 20% of the brain cells pretty much reproduced all symptoms of the disease, including the premature death. Now, what we learned afterwards is that taking out this protein from the inhibitory neurons did not totally inactivate them. They were still functioning, did not kill them. They were still there. What happened is just slightly dampened their function. It slightly disabled them. So what is the big surprise here? Who would have thought that very partial disabling inhibitory neurons very partially is enough to cause a plethora of neuropsychiatric symptoms and premature death? This was possible only by knowing the molecules that define this group of cells, having one protein that we could connect to a multitude of neuropsychiatric behavior. And only with that we were able to really gain some new insights. So we must capitalize on such detailed molecular information and behavioral data as we study circuits. Second question is, how do various cell types connect and communicate? To understand networks, we need to understand points of contact. Again, this is an area where we're just beginning to gain knowledge. While retrograde labeling techniques using rabies virus are successful, we can optimize retrograde labeling techniques and we can develop anterograde techniques. Renaissance disciplines in neuroscience, such as retrograde mapping and optogenetics, relied on virology and microbial biology. We should remain open-minded to what other disciplines have to teach us. Having tools to study specific cells and their inputs and outputs will be extremely important to this project. Third, we need to figure out how to study how cells communicate. This is the most obvious area where we need disciplines such as physics, mathematics, bioengineering, and computational sciences. We need to develop technologies so that we can visualize and record from cells deep within the brain, not just cells on the surface. We also need to increase our ability to record from a large number of cells at a time. Of course, the real question is, do we have to record from every cell within a network? I suspect that a more complete understanding of each brain cell type and their inputs and outputs will help us determine the number we need to record from. Might we learn a lot from recording from just 10 to 20% of neurons? I don't know. We should think about that. I'd like to offer an analogy. Merely owning the Oxford English Dictionary does not enable one to speak English. When we learn a language, we have to learn its basic structures, its rules of grammar, noun declensions, verb tenses, and so forth, along with its irregularities, which is usually helped by understanding its history. In other words, we want to know the principles of how language works so that we can understand what it is capable of. In neuroscientific terms, we want to know the principles of neuronal function, not just recording of their activity over a certain period of time. Fourth, how does circuit activity vary over time based on different experiences and external stimuli, not to mention developmental stage, age, sex, activity time of the day, and disease progression? We need to remember that brain activity doesn't happen in a vacuum. Even if one recorded all spikes from all neurons over the entire lifespan of the organism, one could not make any sense of the data without simultaneously recording all external stimuli and all aspects of behavior. And that would be just for one animal. Although the goal of comprehensive measurement sounds enticing, there would be no end in sight. Furthermore, we know that we lose hundreds of thousands of cortical neurons at a steady state from early childhood till adulthood. This is another argument why we might not want to consider recording from every cell. Many will disappear. A circuit in a newborn animal with limited experiences is vastly different from that of a mature adult animal. Should we consider recording from such vastly different circuits? I think we should consider this for many reasons that we might debate during the next 2 days. But clearly, we need to define not only what needs to be measured and in which cells, but we need to have defined endpoints. I would posit that the map can only be understood if we know the identity of the cells whose activity we are recording, understand their unique biochemical properties, and determine how the activity of each of these cell types changes in response to different conditions. The power of approaching the map, combining molecular anatomy and physiology, goes beyond our ability to interpret activity. It will help us connect our discipline with advances from many other disciplines. For example, We know that there is a lot of human genome sequencing of individuals with neuropsychiatric disorders. We expect that we're going to find many, many genes associated with these disorders. But this will stay dormant and not very helpful until really we connect it to some of the studies we're going to think about over the next 2 days. If we can find ways to develop functional relationships between disease-associated DNA changes, molecules that define brain cells, and activity changes, this will bring us closer to understanding how a circuit might become vulnerable. Biology and genome projects and neuroscience have taught us the value of model organisms. We need to continue to capitalize on this. I'll give you another example from my own work. We studied a gene that is homologous to a fruit fly gene called atonal. In the fruit fly, atonal makes cells, just a few cells, cells of the auditory organ and proprioceptive organ. They are really a cluster of a few cells. But in the mammal, the same gene defines multiple components distributed through multiple tissues of the proprioceptive and auditory system. So here we have an example of a macrocircuit determined by one molecule. So therefore, knitting molecular anatomy, cross-species studies, and connectivity in the overall activity map will have a great impact. Obviously, some behaviors we're interested in would be better studied in humans than animal models. Somehow we need to make the research much more iterative between humans and the lab. There's a wealth of information in the clinic and in patients with neuropsychiatric diseases. Somehow we need to have science penetrate that and really keep going back and forth, but use the information in a much more rigorous way. Take, for example, deep brain stimulation. Deep brain stimulation of cingulate area 25 seems to be helping some depression. But it doesn't help everybody. There's some patients that respond, and some patients with the same stimulations don't respond. What's different between them? Helen Mayberg used MRI on the white matter to study a difference between the responders and non-responders, and found that the responders had activation of white matter pathways linking cingulate 25 to 3 distinct remote areas. She's now testing, she and her colleagues, a new paradigm that better activates a more distributed network. Such studies can guide lab work in animal models for fine mapping of such micro and macro circuits and can then inform clinical research. Last but not least, the success of this project will depend both on the people who receive the funding and on the greater scientific community that will not directly participate in this project. Thus, it's really critical that all the data generated from this project goes public as soon as they're generated and be made accessible and, more importantly, user-friendly so that everybody can use them. This is why the genome had such an enormous impact. Data came online immediately, and the larger scientific community put it to great use. In fact, most of the significant insights came from the greater community and not the data generators. The data generators basically were really performing an altruistic service for the larger community. So we must adapt this model and have a very well-designed brain browser that every scientist can use. Thank you.